Sound should be going through no problem. I'm going to start monitoring the live chat, so anyone who has questions or any particular things in the study guides that you want me to review, just let me know in the live chat and I will take a peek. Okay, cool. Looks like we've got some questions coming in. Okay, so how to complete a frequency table from the population genetics. Okay, and then another example of convergent and divergent evolution. Okay, really good points here. So when we're doing the frequency table, let me go ahead and open up a document here real quick that I can show you guys with the frequency table. Let me see how many there'd be. Let me make sure that this is visible real quick. so let's see what else. Okay, so it looks like there's some audio problems again. But now that it's back on my face, you should be able to hear me. I'm using a new program, so I appreciate you bearing with me with all of these technical difficulties. I should have changed the audio on all of them now, so it should be working if I change screens. I'll make sure that I'm still monitoring that for next time so that you don't just see numbers appearing on your screen. And that'll hopefully be a little bit better. Okay, I'm going to check with all of you real quick. And we'll hope for the best on that. Okay. So for now, I am assuming that I can be heard okay. Let me double check that this one is also correct. Okay, it looks like all of these are now going to be okay. Okay, so we wanted another example of convergent and divergent evolution. So let me pull up some examples real quick. Oh, I got a great example. Oh. So I'm just pulling up some pictures real quick. Awesome. Okay, you guys can all hear me. That's delightful. Sweet. Volume. Yes, it is sweet. I do what I can. All right. So I'm going to pull up some photos here. As I've been saying, I've been trying to grab those. Let's see if I can switch screens without absolutely messing everything up. And we'll hope for the best. Oh, look, it's an infinity of my screens. Okay, it looks like my mic is still working now that I've switched kind of what my feed in is working. So let's hope that it stays that way. Okay. So I'm going to do some examples of quick convergent divergent evolution. Is this idea that different things, right, if they undergo the same evolutionary pressure, are going to end up looking the same. Okay, so there was evolutionary pressure, right? There was natural selective pressure pushing for the evolution of wings. And so wings have evolved a couple different ways, a couple different times. So here we have a bat wing, and a bat wing is going to actually give them the power of flight through these stretched out phalanges. They're going to have this membrane kind of across their finger bones, versus birds, which have still kind of this similar arm bone structure, but now they have feathers. So they're flying, but it's a very different kind of flight. But these two are still pretty similar, right? We can see there's one big arm bone here, there's two smaller ones in our forearm right there, and then we have kind of this fused section of phalanges in the bird, and the phalanges are all spread out in the bat. However, for an insect, insects have their legs on the bottom of their body. Okay? So their legs are underneath them, and their wings are actually coming off of their back. Okay, so for these wings, which belong to a dragonfly, the dragonfly is going to have 6 legs underneath it and then 4 wings coming off of its back. So it actually has 10 separate appendages. And so the wings, while they do the same thing, right, the insect and the bat are not closely related just because they both fly. Both the bat and the insect evolved wings separately. This is an example of convergent evolution. Okay? Evolution converged on structures that have similar functions for different species. I'll say that again. Evolutionary pressures converged on structures that have similar functions for 2 different species. Okay? This is convergent evolution. Both shapes are converging on one type of function. Okay, so the other example that we saw in class was with the what was it, the seal and the penguin and the tuna, right? They all have torpedo-shaped bodies, not because they came from a torpedo-shaped ancestor, but because they all needed to be very fast in the water. Okay, so an example of divergent evolution, okay, is here where we see all the different arm bones. Okay, so a human and a horse and a cat and a bat and a bird and a whale all came from the same common ancestor. So these are going to be homologous structures where we all have this one kind of big upper arm bone here, right? And this, we have it in the human, in the horse, in the cat, in the bat, in the bird, and in the whale, right? Even though it's been modified, right? In the cat, it's kind of a little bit thicker. In the horse, it's very short. In the bat, it's very thin. In the whale, it's incredibly small, right? So this bone has kind of been modified. If I gave you kind of the basic parts, and I said, hey, make something that's really good at grabbing, you'd probably arrange the bones kind of in this fashion. But if I said, hey, use the same bones, you can modify them a little bit, but now I want you to make it so that you can, so that you're really good at standing and running, you might arrange them like this. If I had you arrange the bones and kind of just tweak them a little bit, right? You can't suddenly make a bunch of brand new bones, but you can modify the existing structures for something that makes the animal walk very quietly and make it good at sneaking up on prey or climbing trees. You might make something like this. If you want to make something that flies through the power of jazz hands, you would make this. If you wanted something that's really good at pushing water, you would elongate these phalanges and shorten all the other bones and then put some kind of membrane around the entire thing. That's now going to be really good at pushing water. Okay? So homologous structures are if evolution kind of had the same basic template and then had to use it and shape it to make a bunch of different things. Okay? So all of these have very different functions, right? Grabbing, running, climbing trees, flying, also flying, swimming, very different functions. But the anatomy is the same basic pattern, right? This example here, this anatomy and this anatomy are incredibly different, right? The anatomy of a bat wing and the anatomy of an insect wing are so insanely different. First of all, bats have bones, right? Bats also have fur, and they have this kind of membrane across their bones. Insects do not have bones, right? They don't actually technically have fur either. They're a completely separate evolutionary lineage. So the structure does the same function, but they got it separately. Okay, so that's the explanation of convergent and divergent evolution. A quick answer for the table. So for the table, because that's when I was doing it silently and you guys are just watching a bunch of numbers on the screen. For the table, the values that I picked, the like 2, 3, 4, 6, whatever, those are just arbitrary numbers that I picked to say like, oh, we're working with pairs. And that's something that if you were doing the experiment in lab, you would have made those pairs yourself. But if you are just given some numbers, you should be able to work through it as well. If we have some time at the end, I'll go back through that Punnett square example, this time with sound. Okay, question about how do we order the taxons on a phylogenetic tree? About kind of which ones go on the top or the bottom. So for phylogenetic trees, it's not so much top or bottom that really matters, because I could also turn the tree, instead of going this way, I could turn it this way and it would still give you the same information. So as long as I have for the phylogenetic tree, as long as I have the shortest lines for the things that evolved most recently, that's going to be an accurate depiction of what actually happened, right? But we've been showing it kind of the way that we've been showing it, we've been showing the more recent species on what we would call the bottom of the tree, and the more old species closer to the top. And if you want to stick with that, that's fine. Technically, if you flip the entire tree like this, you're still showing the same information. So if you're still showing the same information, that's what's really important to get across in those trees. So, let's see what else we have here. Oh, this is exciting. I can pop out the entire chat and look at it myself. Oh, that's lovely. Beautiful. Okay, can I explain parsimony? Yes, I can. Yes, I can. Also, please keep the questions coming. If you put them in the chat, I'm constantly going back into the chat and looking to see what people have been asking to make sure I get to all the questions that I can. So let me go ahead and pull up the right slides here. And it looks like the slides are now working with sound. Chapter 15, now with sound. All right, so we're going to look at phylogenetic trees here. And we looked at this example in class very briefly and then said, that's disgusting, we're not going to do that. So we'll look at this one briefly, but mostly looking at what we're trying to figure out here is that parsimony means that there's the least number of changes. So in our example here, okay, we said that amnion evolved right here because all the creatures that came after this point, the iguana, the platypus, the kangaroo, the beaver, all have amnion, right, which is a little protective coating around the egg. We then can group these three because they all have hair and mammary glands. And then we can group these two because they both actually gestate their young, so they don't lay eggs. And then we can separate the beaver out by itself because it does a long gestation. The kangaroo doesn't do a very long gestation. It actually gives birth and the baby goes to live in a pouch because a kangaroo is a marsupial. This is one possible explanation for how this tree could work. Another possible explanation is that this ancestor way back here also had hair, but then we lost hair here for the frog, and we lost hair for the iguana, and then gained it back, or rather kept it for all of these groups, okay? Which basically means that that's a very complicated explanation, right? That's saying that a lot of things would have happened. So that's saying that there was a common ancestor with hair that then had to lose hair to then evolve into a frog, that then also had to lose hair to evolve into an iguana, and then had to keep the hair to evolve into all of these things. What's a lot more likely is that this ancestor probably didn't have hair. Hair probably only evolved once. Okay, so hair probably only evolved once, and it probably evolved right here, because these three things are the things with hair. So with this very scary looking slide, we can look here and kind of look at our different explanations. Okay, so I'm going to walk you through the slide real quick. So we have our different species of birds. So we have species 1, 2, and 3. And then we have three possible ways that we could orient the tree. Okay? So we have either species 1 is more closely related to species 2 than 3, or it's 1 and 3 that are more closely related, or it's 3 and 2. Okay, so these are the three possible ways that we could set up the tree. And then we also have some sequences here where we're able to figure out that the ancestral sequence of DNA at a couple different sites actually goes A, G, T, T, and then we can see where we have differences for each of the species. Okay, so this one for species 1 at this site has C instead of A, meaning it's different than the ancestor, but this one has A, so this one is more closely related to the ancestor, and that's what we're showing right here, is we're saying, okay, in order for this to go from the common ancestor and then over, to go from the ancestral sequence over, there's one site, and this site has to change to C, and then we also, let's see here, which is the order? Oh, this is just for the first site. Okay, so the first site for this one has to change to C, right? Because in the ancestral sequence it's A, so in order to get over here, it'd have to change to C, right? To get over here, it would have to change right there, and to get over here, it'd have to change at both of these places. It's not quite as important to be able to follow exactly where these marks are put, because this one is actually pretty complicated. Okay, but I'm just going to kind of jump down here to this results section, where we look and see, okay, if we arrange the tree like this, okay, we're trusting that our data is good, so we're trusting that we set this up correctly. If we set the tree up like this, where 1 and 2 evolved most recently, and then 3 kind of split a while back, this leaves us with 6 different events. So the DNA had to change here, here, and here, and here, as well as there and there, in order to give the species that we now see. The other options have 7 events. 7 different changes had to happen. Okay? So parsimony would indicate for these trees that either the DNA sequence had to change 7 different times for this evolution, for this evolutionary pattern to be true, or it had to change 6. And the way that it works usually with evolutionary trees is whatever amount of times you have to change, it's the least number of changes, the least number of change events that have had to happen is likely the most correct. Okay, so parsimony would say, okay, if you have to change six things instead of 7, this is most likely what actually happened. That's the big takeaway in figuring out how we actually know that our trees are correct, right? Because we're working here with our example, we're just working with traits that we can actually see in these creatures, right? We can actually look at their eggs and ask, do they have amnion? We can look at the animals and say, do they have hair? Do they have mammary glands? Do they gestate their young? And then we arrange them like this. And it's very possible that our tree could be incorrect. Phylogenetics is a constantly expanding and changing field where we kind of arrange things and then figure out with some really intense research that it's actually not quite what we thought, and so then we change the trees a little bit, and that's actually currently going on within the Eukarya domain. We're figuring out some other stuff about that tree to figure out how it's correct, but we usually stick to this rule of parsimony, which is that it's the most simple explanation, right? Things have to evolve the fewest amount of times is going to make the most sense. Okay, so for the, there's also a question kind of going back to, let me switch here real quick, going back to the table. With all the different frequencies. So for the frequency of allele, it doesn't always have to be 200. So the question was asking for the frequency of the allele when we're trying to calculate what's the frequency of little l or what's the frequency of big L or something like that. We've been doing it with nice round numbers, so we've been giving you numbers like there's 100 total individuals and there's 200 alleles because it's much easier to work with. We could give you an example where we say, okay, there are 65 total alleles in the population. This many are big, this many are little. What's your frequency? And you just have to divide however many you have over the total. We tend to give you nice numbers so that it's less about the math and more about if you actually understand what you're doing. So with how we've been doing it, with the way that we've been working out the math, it has always been 200, but it doesn't have to be, right? Because if you're working with a population of like 13 individuals, if you're working with a population of 13 individuals and each of them is diploid, you're going to have 26 total alleles. Okay, so if you're working with a population that has 67 individuals, you're going to be working with a population of, I have a calculator right here, then you're going to be working with a number of 134 total alleles. But we've been giving you a population of 100 so that your total number is 200. So I would always check and figure out how many individuals you have total, and then however many individuals you have, double that, and that's how many alleles you have. So I emailed my students and said for lab 12 that you need to complete the graph for plotting the frequencies for each generation. So the way that you would do that is you let your professor go grab her lab manual so she can show you. All right. Handy dandy. Also, while I get this open, can I just say, technology is so cool. The fact that I can just live stream from my house right now and just give you guys information about science, it's just so awesome. Okay. I feel like the young, hip version of Bill Nye. All right. Let me see if I can write this big enough. This is going to be Okay, so I just made up some data to work with real quick. Okay, so I'm working with this data here. So for a non-random predator, okay, I'm going to say that I started with 0.5 frequency of our little R allele, and then it went down over the next generation. So it went down to 3, and then it went down to 0.2, and then it went down to 0.1. So now what I'm going to do is I'm going to figure out my axes. Okay, so I'm going to think about how this frequency of this allele and how these generations are related. So as generations progress, will that change allele frequency, or will allele frequency change the generations? And so through that phrasing, I can figure out, okay, it's going to be as generations progress, that's going to change the frequency of the allele. So that means that frequency is going to be my dependent variable. Okay, so I'm going to put my frequency right over here. I'm going to put it on my y-axis, and I'm going to put generations down here. Okay, and I have my original population at time 0, and then I have generation 1, generation 2, and generation 3. Oh, that doesn't work out nicely. Oh, why are these squares so ugly? Okay, so here's what my graph is going to look like now. Okay, so now I have my frequency here, and I say, okay, this middle part is 0.5, so it could either go up or down from where it started. And then here it's going to go down to 0.4, 0.3, 0.2, 0.1, and then 0. And so here I have, as my data says right here, okay, at generation 1, I have a frequency of 0.3. And then at generation 2, I have a frequency of 0.2, and then at generation 3, I have a frequency of 0.1. It's really hard to do this in the camera, and so I'm just putting my pencil everywhere right now. Okay, so what we generally see here with this graph, if we connected all of these dots, is that we actually see this kind of pretty consistent downwards sloping line. Okay, so we can see that the frequency of this allele is consistently, through time, this side of the graph, the frequency of this allele is going down over time. Okay? And this was another question on the quiz. If the frequency of an allele is going down over time, that means that it's not beneficial to the population. It's getting selected against, right? Because if there's an allele that is causing, what's a really good disease? What's a good disease? Let's say sickle cell anemia. Or let's say it causes a disease, it's like, oh my goodness, early onset leukemia. Okay, so let's say early onset leukemia, right? Most of the individuals that are born with that allele, if you have like half your population born with early onset leukemia, everyone that's born with early onset leukemia is going to die before they hit reproductive age. So that allele is going to get very quickly kind of wiped out of the population. And so in all the coming generations, that allele is going to go down in frequency because it's not making it into future generations. Consequently, if you see the frequency of an allele going up over time, it's usually being selected for. It's a beneficial allele, right? It's a good allele. It's helping you. So if you have an allele for, let's say, night vision, right? Increased vision at night so you can see really well in the dark. And all of your prey, right, lives at night. Usually you hunt at night and you become nocturnal. Then if you have night vision, you're going to be really good at hunting. If you're really good at hunting, you're probably going to get more food. If you get more food, you're going to probably be bigger and fit, right? If you're going to be big, you can probably fight off all the other males, and then you can mate with a female. And then if you're mating with a female, that means that you get to have offspring, which means those offspring are now also going to have night vision. They're going to be better than the other offspring. I'm thinking of probably like jaguars right now. And then those offspring are going to have night vision. And when those offspring have night vision, they're also going to be able to hunt really well and get a lot of prey and be very fit and have a lot of babies. So we're going to see this kind of increase of night vision over time, which is going to be beneficial for the population. It's something that's being selected for. So if an allele is going up over time, it's beneficial. It's being selected for. If it's going down over time, it's being selected against. That being said, the other interaction that we had wasn't just selection, right? We didn't just see the change of an allele frequency with selection over time. We also saw it with genetic drift. So the random predator and how it kind of fluctuated from like 0.5 to 0.48 to 0.52 to like 0.47, there's little fluctuations over time. It's random, right? You're not necessarily going to stay at exactly 50% of your dominant and 50% of your recessive allele. It's very positive that your allele. It's very possible that your alleles could fluctuate a little bit over time, and that's drift. It is possible that alleles could randomly just drift out of the population, but most likely, if you're seeing something like incredibly decrease over time, especially like 3 generations, that's not a lot of time. Right? We've been talking about speciation happening over thousands of generations. So if you've seen an allele drop from like 5 to 2 in 3 generations, it is insanely bad for the population. It is being selected against at an incredibly high pressure, right? Consequently, if you've seen an allele jump from like 0.5 to 0.9, that's really, really good for the population, right? So there's a lot more of it over time. And so that's the idea behind kind of graphing, and why do you want to graph it? Because then we can look at the graph and figure out, is this allele actually good or bad for the population? Or is it neutral, right? If it's staying the same, then it's probably not really it's not being selected for or against. Oh my goodness, okay. I don't know who's trying to calculate p squared and 2pq, because I do love me some Hardy-Weinberg. Oh man, do I love Hardy-Weinberg equilibrium. But I'm going to go ahead and do you all a favor and not get into that. We're not going to talk about Hardy-Weinberg equilibrium. If you want to grab me at some point after class during office hours and have me give you like a cool sit down about how Hardy-Weinberg equilibrium works, and the way that like mathematics and evolutionary biology and population genetics had this like really awesome cool love child that's just the field of like math and evolution, we can totally talk about it. But I will spare you all from that right now. Okay, some examples of macroevolution. Oh man, okay, examples of macroevolution. So macroevolution is speciation. So anything that we can consider a species that's a speciation event. Okay, so that's a macroevolution type event. So anytime that something became a different species, so like humans would be an example, and like dogs and cheetahs and elephants and lions and sagebrush and chaparral broom and laurel sumac and paramecium and hydra, all of those very different species, those are examples of macroevolution. Okay, so anytime anything becomes a new species, that's macroevolution. A really, really big scale, something becomes a new species, or you get a bunch of different species. A really beautiful example of macroevolution is Darwin's finches, where you have one ancestor and then you have this huge explosion of all these different kinds of species of finches living on the Galapagos Islands all together. That is a great example of macroevolution. Right, we start with one species, and over a relatively short amount of evolutionary time, which is still a long amount of regular time, you get all of these different species. Okay, so anytime that you have something become a new species, that's a macroevolution event. And something that's important to remember about macroevolution, even though I say like, oh, it's very short evolutionarily, we're talking thousands of years, potentially millions of years. Right, we're looking at our geologic time scale today, and we're seeing that big benchmarks in geologic time happen in the scale of like billions of years. And for a human, it's hard to even wrap our head around 100 years. So when we try to expand and go into thousands or tens of thousands or hundreds of thousands of years, it kind of gets away from us pretty quickly. Okay, I'm going to open up some study guides here. So I've had some requests on those. Let's get these open. Yeah. Okay, so if you have a question of like, is this going to be on the exam, definitely double check with your study guide. I'm going through the study guides right now and looking to see if there's anything on Hardy-Weinberg equilibrium, and there's not, because Hardy-Weinberg equilibrium it's definitely not something to mess with. So to one of the questions of like the Hardy-Weinberg equation is not on the exam. No, we are not. We have not presented the Hardy-Weinberg equation. We haven't even shown you the equation. We haven't talked about how to use it or what it does or when you can use it or when you can't use it or how to figure out if you can use it. It's so cool. I'm dying that I can't explain it right now, but no, it's not going to be on the exam. Okay, so can I go over number 5 on the chapter 15 study guide? Compare the methods for categorizing living organisms into the 3 domains of life. Yes, I can. So let me pull up some slides. Oh, this is so fancy. I'll just jump right back into the slides. All right, so let's do a little quick review, kind of orient ourselves with where we are. Okay, so here's our 3 domains of life, where we have domain bacteria, and then we have domain archaea, and then we have domain eukarya. The way that we classify organisms into these 3 domains of life are using this systematics that we have in place. Okay? So our domains down here, we have eukarya and bacteria and archaea. If something falls into one of these domains, we can then further classify it into something like a kingdom, and then we can get even closer and classify it by a phylum. We can go into class, into order, into family, and genus, and species. And let me pull up another site for that. Give me 1 second here. I'm just pulling up a... here we go. All right, I'm going to pull up one of my favorite plants, because plants are so cool. All right, so here's a Wikipedia page on the Nepenthes, the Nepenthes genus, which are the pitcher plants. So you might have seen these in our botanical building when we did the walk in Balboa Park, and these are carnivorous plants, so they actually eat things. They are considered a producer and a primary consumer, which is super cool. So in the Wikipedia page here, we can see all of this cool information, but for any species, we can go over here and look at our classification. So here it tells us, okay, so we're in the kingdom Plantae, and then the weird thing with Plantae is we don't have a lot of these kind of intermediate classifications, right? Like order, family, all of those, or phylum, class, and all of those. And so we kind of jump down here to order and see it's in the Caryophyles order, and then it's in the Nephthemceae family, and then its genus is Nepenthes. Okay, so for every single species we have this, and let me see if I can pull up another one real quick here. That wasn't very good. But for any species that you pull up on Wikipedia, it'll have that classification kind of over to the side. Ah, this one is much better. One of my other favorite creatures, the honeybee. Okay, so here again, kingdom Animalia, phylum Arthropoda, class Insecta, order Hymenoptera, family Apidae, subfamily Apinae, tribe Apinae, and then the genus Apis. And then under here we have all the different species, again using that kind of 2-word name. So we have Apis dorsata, Apis florea, our most common, Apis mellifera. It's one of my favorites. Okay, so this just kind of tells us what different grouping it's in. So all of the arthropods, so the things in Arthropoda, are going to look more similar to things within Arthropoda versus things in the phylum Chordata. We're in the phylum Chordata, and the phylum Arthropoda are going to be our arthropods, so we're going to see like crabs and spiders and insects and a lot of other things in that group. Same with things in the class Insecta. Right, so this bee is going to look more similar, it's going to have more in common evolutionarily with other things in the Insecta class than things in other classes under the Arthropoda umbrella. And let's see here. So I'm trying to figure out if I fully answered that question. So the methods for categorizing living organisms according to the 3 domains. Yeah. Yeah, I think that's pretty good. Basically, that we have different defining factors that we can use to kind of say that something is a bacteria versus belonging in archaea versus belonging in eukarya. And so that's kind of that's our idea behind systematics, is that we can name things to understand how they're organized. Oh, good. Okay, a couple different things that are going to be really good. It'll probably take us to the end of our time. So polyploid speciation, Lamarck's idea of evolution and Darwin's, and then sexual dimorphism. Okay, so there's a couple of these that I can get to pretty quickly. So sexual dimorphism is when the two sexes of a species look different. So we see sexual dimorphism in a lot of birds, like peacocks. So with peacocks, we have the male peacock, and they're very big and showy and blue, and they have like a cool little mohawk, and they've got their big fantail. And then the peahens, the females, are small, not a long tail, kind of a brownish color. Same way, we also have sexual dimorphism with lions. So with lions, the males are generally, they have this really big ruff around their neck. They have a lot of fur. It's kind of a darker color, and the lionesses don't have that. So sexual dimorphism is more of a difference between the sexes. It doesn't have to necessarily do with directional selection, because we don't really know necessarily how they got those traits. If you're asking about non-random mating, so if you're asking about sexual selection, sexual selection could be directional, but it could also potentially be disruptive. And it could also be stabilizing. Okay, so here's how that would work. Let me see if I can pop up the slides that have that real quick. Really excited that we get to do one about the different types of selection. It's one of my favorite concepts. Oh, perfect, it's right here. All right. So here we have our chapter 13 slides. So we have our patterns of natural selection. Okay, and so let's say for this selection, it's not natural selection. It's not like the environment is saying that you need to be a certain color. What we're going to say here is that the female is choosing the color. Okay, so we're going to say that this is sexual selection, right, where the females are choosing and they pick what they like. For stabilizing selection, if the females choose this intermediate, that's stabilizing selection. Okay, stabilizing selection is, when the intermediate is favored over the extremes, right? We have our gradient of phenotypes here. So we have one extreme, which is fully white. We have another extreme, which is fully dark. And then we have this intermediate, this is gray color. Stabilizing selection selects for the intermediate and against the extremes. And so all the extremes over here are going to die off, and we're going to end up with everything basically being this intermediate phenotype. So if the ladies like the gray mouse, then it'll be stable. If ladies like the darkly colored mice, then it'll be directional selection, because they'll only mate with the darkly colored mice, and then their babies will all be dark, and their female daughters, which is the only kind of daughter you can have, their female offspring will also have the preference for darkly colored mice, and it'll be directional, and then it'll pick one extreme over everything else. Okay, so it's selecting just one of the extremes. If some of the ladies like the white fur and some of the ladies like the dark fur, that would be disruptive selection, because you'd be selecting for both extremes. Okay? So sexual selection is a mechanism of evolution. And usually we see these types of natural selection usually based on habitat, so usually based on the environment that they're in. So they are two separate concepts, but you can kind of connect them. One of the big take-homes from this is that stabilizing selection selects for the intermediate and against the extremes. Directional selection selects for one of the extremes. Disruptive selection selects for both of the extremes. So understanding the definition as well as what these graphs look like is going to be very important. Okay, so make sure that you understand how to do that. Other quick thing that I can get to right here is the difference between Lamarck's idea and Darwin's idea. So Darwin's idea of evolution is the idea of descent with modification. So you have some kind of variation, And then your offspring is also going to get that variation, and then they're going to pass that variation on to their offspring. So if you're a bird and you have a beak that's slightly bigger than the rest of the birds, then your offspring are also going to have slightly bigger beaks, and maybe one of your babies has an even bigger beak than all of its siblings. And then that bird does really well because big beaks happen to be favored. Maybe they can crack seeds open more easily. And then that one is going to have a lot more offspring, and then that one has a lot more offspring. It's going to have babies that just happen to have bigger beaks, and so on and so forth. So this idea of you're going to have babies and your babies are going to look like you regardless of what you do in your life, right? If the bird lost its beak, right, if you go to breed a duck that has had its bill, whatever, cut off for some reason, its babies are still going to have bills, right? If you have a bird with no beak and it has babies, all of its babies are going to have beaks because it's genetically programmed in the gametes of like, your baby should have a beak. Lamarck's idea of evolution was this kind of you mess with your genes during your lifetime idea. So one of the ideas that he had was with giraffes, right? And so Darwin's concept of giraffes is there's a population of giraffes. Some of them have slightly longer necks, some of them have slightly shorter necks. The ones with longer necks are going to be able to reach higher in the trees. They're going to have babies with longer necks, and they're probably going to have more babies than the ones with short necks. Those babies are also going to have long necks, and some of them might even be longer than their parents. They're going to be able to reach the higher leaves, so they're going to have more babies than the ones with short necks, and then their babies might have even longer necks, and so we see this very gradual change of kind of short to very long necks over time, not because the parents were stretching their necks out really hard during their lifetime, which is Lamarck's idea. Lamarck's idea is that the giraffes were stretching their necks during their lifetime. So the parent was stretching their neck and stretching the neck and stretching the neck so much that when they had a baby, the baby also had a long neck. Which when you phrase it in other ways like muscle tone or hair color or losing a limb, like if you work out all the time and you get super swole and you drink your protein, your baby is not going to come out looking like Arnold Schwarzenegger, right? Just because you're a bodybuilder does not mean your baby's going to come out looking totally ripped. That would be awesome, but that's not how it works. If you dye your hair crazy colors, right? If you dye your hair purple, your baby's not going to come out with purple hair. The other one, if you lose a limb, right? If you lose an arm, your baby's not going to come out without an arm, right? You can modify your body during your lifetime in a lot of ways. Those modifications don't get passed to your kids because they're not heritable. Okay, so what Lamarck kind of missed was the idea of what exactly is heritable, right? What exactly can you pass on to your offspring? Darwin understood only some things are heritable. Not everything is heritable. And what you do to your body in your lifetime is not heritable. Okay, so that's the major difference between Lamarck and Darwin. Last big one is polyploid speciation. So how can speciation occur? And we said that there's two different types, so I'll go over both of the types of speciation real quick. Let's get these slides up. Shoop. All right. So modes of speciation, we have two different types of speciation. We have what we call allopatric speciation, which is they are physically separated. Okay, there's some sort of physical barrier in here, and now they're going to be separated from each other. The other type is sympatric speciation, where they're literally turning into different species right on top of each other, which is a little tougher to understand, right? Because like if you're right on top of each other, how are you not exchanging genes? How are you not interbreeding? How is gene flow being uninterrupted if you literally live in the same pond? And so that's where we got into some ideas about how you can actually interrupt gene flow. Allopatric speciation, genes can't flow across this canyon between these two chipmunks because the chipmunks physically cannot get across the canyon. Okay, so gene flow is interrupted, which lets this population become different from this population, and over long enough time when they diverge, they're going to turn into different species. For sympatric speciation, okay, we have this example about sexual selection, but we're going to focus in on this one about polyploidy, which has to do with the fact that plants don't really care about things in the same way that humans do. Plants are super hardcore and awesome. And one of the things that they don't really care about is how many chromosomes they have, right? So how many chromosomes a human has can be really, getting the wrong number of chromosomes can be very detrimental. for a human. For a plant, not so much. So the way that a new species can evolve right next to another species is potentially through polyploidy. Okay, so poly meaning many, and ploidy meaning how many sets of chromosomes do you have? So here we have the wild version of wheat. So we have wild triticum that has 14 chromosomes. And we have our domesticated triticum that also has 14 chromosomes, but these are different enough that if you breed them, you get a sterile hybrid. And this sterile hybrid is sterile because it only has one copy of the important information. So this one has 2 copies of the A chromosome, and this one has 2 copies of B. This one only has 1 copy of A and 1 copy of B, so it can't undergo meiosis, right? It's got 1 A and 1 B, and you can't divide 1 into 2 evenly. So this one cannot undergo meiosis, and it's going to be sterile, unless by some fluke cell division error, and then it self-fertilizes, it doubles, it effectively gives itself a second set of chromosomes. And that's what happens. Okay, so with this Triticum turgidum, this now has 28 chromosomes. This 28 chromosome plant cannot interbreed with this 14 chromosome plant back here, right? These two are no longer the same species. And they could be growing right next to each other. But now they're not the same species because this one has a different number of chromosomes than this one does. So even if they did breed, they probably wouldn't be able to produce any kind of fertile offspring. Okay? And then it actually went one step further because it's plants, so of course it did. So what happened in the wild is this wheat here with 28 chromosomes hybridized with this other wild species that had 14 chromosomes, and we saw kind of the same thing that happened the first time. We get a hybridization event where our hybrid is sterile. So now we have 1 copy of A, 1 of B, and 1 of this new chromosome, this D chromosome. So we got a new chromosome here. We got some new chromosomes from this one. And now we've got 21 chromosomes, right? We can't divide these evenly, so it's not going to be able to reproduce. It's going to be sterile. And then because it did cell division wrong and it self-fertilized, we end up with 42 chromosomes. So our end product is this Triticum aestivum. That's right. So the Triticum aestivum has 42 chromosomes. This is a completely different species than either of these two parent species. And that's what we're talking about with sympatric speciation, is we're saying you're making a new species in the same physical place. You can grow this plant right next to this plant or right next to this plant, and they're not going to be able to have viable offspring because they're different species. The way that they became different species is through polyploidy, okay, for the fact that they can just, and sometimes they do just double the number of chromosomes they have, because plants are weird like that. Okay, so this is one of the examples in that plants can sometimes double the number of chromosomes they have, sometimes on multiple occasions, and that will make them turn into a different species than what we started with. Right? Because up here we have 14 chromosomes, and down here we have 42 chromosomes. Very different species. Okay. So that's the example that we went over with polyploidy. All right. So, let me go ahead and check something out real quick. Cool. Okay, it looks like a couple of you are still hanging out here. So if anybody has any last minute questions, I'll hang out for maybe another 5 minutes or so, because we did have some sound issues in the beginning. Also, if you would like me to go over kind of that paired number Punnett square thing from Lab 12, let me know, and I can spend a little bit of time going over that. But I'll just hang out for a couple minutes and see if anyone has any other questions. And if not, then I will send you all on your way and wish you the best of luck studying. Ah, yes, the Punnett square. Okay. I will go ahead and, yeah, I'll do the Punnett square real quick because you do have your phylogeny at home to kind of help with some of the cladistic stuff. Okay, so last thing I'm going to do is the Punnett square. All right, let's go back over here to this document. That is my face. That is wrong. Okay. So we're going to go back over to this Word document here. Okay, so I'm going to backtrack a little bit, and we'll work with some new numbers, and we'll talk about how we originally got these numbers in the first place. So bear with me for a second while I set this up again for us. Okay, so the way that we set this up originally is we're trying to figure out what's going to happen in the next generation. So we're trying to predict the offspring of a bunch of mating pairs. So we're trying to predict what's going to happen with the next generation with regards to genotype. So first we list all possible mating pairs that we could have over here, right? So we could either have a double dominant mate with another double dominant, or a double dominant with a heterozygote, or a double dominant with a recessive, or two heterozygotes together, or a double recessive with a heterozygote, or two double recessives together. Those are all possible combinations of the mating pairs as parents. And we said for each mating pair, okay, there's something special about these pairs, each mating pair has 4 offspring. So we fill this in for one mating pair. So we say, okay, if we have one pair and they have 4 babies, what are the genotypes of their babies going to be? And we can use a Punnett square to figure out, okay, all 4 babies are going to be this genotype. And we did that for the next cross, and we find out, okay, if you do this cross here, if you cross a double dominant with a heterozygote, half of your babies are going to be double dominant, and the other half are going to be heterozygotes. If you cross a double dominant with a double recessive, all of your babies, which in this case is 4, because each pair has 4, all of your babies are going to be heterozygotes. If you have a heterozygote with a heterozygote, you're going to get 1 double dominant, you're going to get 2 heterozygotes, and you're going to get 1 double recessive. With this, with these last 2 pairs, if you do a heterozygote and a double recessive, you're going to get half are going to be heterozygous and half are going to be recessive. And then for these 2 double recessives, all 4 are going to end up being double recessive. Okay, so this is for one mating pair. Okay, so we do the following chart for one mating pair. And let's choose the following chart is for data we collected in the field. And so I'm going to say that I just went out into the field and I happen to have a population of these on my kitchen table, so we're very lucky. And I just looked at these and I found out that we have 3 of these pairs, because I'm actually counting them. So I'm either counting them or I'm telling you what they are. So we have 3 of these pairs, we have 2 of these, we have 4 of those, we're going to have 7 of these. Tori has to do some math so that she doesn't mess this up again, like she did last time. Oh, the computer. Oh, it is right. Okay. And then we're going to have 6 of these, and we're going to have 3 of these. Okay, and I figured out how many mating pairs we have because I just went over and looked at how many mating pairs I had, and this is the information that I got. So now I'm going to change this table and figure out how many offspring I have for each cross, because with one pair I got 4 offspring that were double dominant. But now I have 3 pairs, so I'm going to say with one pair, normally I get 4 offspring here, but I have 3 pairs, so I'm going to multiply it by 3 and say, okay, now I get 12 total offspring because I have 3 pairs over here. If I have 2 pairs, I'm going to multiply this by 2. Okay, so I'm going to get 4 of these individuals. This one's also going to get multiplied by 2, so I'm going to get 4 of those. I have 4 of these pairs, and normally with one pair, I get 4 offspring. But if I have 4 pairs, I'm going to get 16 offspring. If I have one pair, I usually get 1 of these. If I have 7 pairs, I'm going to get 7 of these. If I have one pair, I usually get 2. So if I have 7 pairs, I'm going to get 14. Okay, and same thing over here, I'm going to get 7. And then for this one, I usually get 2, but now I have 6 pairs, so I'm going to get 12. And for this one, I'm going to get 12. And this time I found 3 pairs, so this one is going to be 3, it's going to be 12. Okay, so that's how you figure out these numbers of how many individuals of each genotype you get, is you just say, okay, how many is it for 1 pair? Okay, so I know how many it is for 1 pair. Now how many is it for 7 pairs, or 8 pairs, or however many pairs? If you wanted for these 4 pairs, you could do a Punnett square for each pair and then count the total number of squares that you made. Or you can do it this way, which is shorthand, and it's a lot faster, and it's a lot simpler, and there's a lot less Punnett squares. So now we can add up our total individuals for each genotype. Okay? And so we can see here we get 12 double dominants from this cross. We get 4 from this cross, and we get 7 from that cross. Okay? So that gives us a total of 23 of these double dominants. For our heterozygotes, we get 4 from this one, 16 from that one, 14 from this one, and 12 from this one. So we get 4, 16, 14, 12. Okay, so we get 46 total here. And then over here, we get 7, 12, and 12. Right? 7 from this cross, 12 from this cross, and 12 from that cross. Okay, which gives us a total of 31 individuals. And then if you wanted to figure out how many alleles you have, right, we could do that too. Okay, so we could figure out how many alleles we have. We can look at this big L allele. We can say, okay, the number of big L's is going to be equal to the number of homozygotes. I'm actually just going to shorten. I'm going to say the number of LL individuals times 2. Okay, because these big L individuals have 2 of these L alleles. Okay, so I'm going to need to know the total number of those times 2 plus the other type of genotype that has this big L is this big L little l heterozygote, and they have 1 of these big L's. Okay, so I'm going to find the number of big L little l individuals, and I'm not going to do anything to it, okay, because this has 2 alleles, 1 big, 1 little. So if I want to know how many bigs there are, there's 1 big L allele in each heterozygote. Okay, and so for little l, it's going to be the same. I said little l. Little l is going to be the number of little individuals times 2 plus the number of heterozygote individuals, because they only have 1 copy of little l. Right? I double this little individual number because these have 2 copies of little l. And so now I can look at our data and I can see, okay, I have 23 of these big combos, so it's gonna be 23 times 2 plus however many of these we have. Okay, so that's going to give us 92 of those. And then for this one, I'm going to have 108. Okay, so I'm going to get 31 times 2 plus 46 is going to give me 108. Okay. So now I can actually look here. So I can look at this and I can see, okay, I have 92 big L's, plus I have 108 little l's, is going to give me 200 total alleles, because that's just the way that the numbers came out, right? If I give you a different number of mating pairs, you would have ended up with a different number of total alleles. Okay, but I ended up here, if you add 23 and 46 and 31, that gives you 100 individuals. So you have 200 alleles. So if I wanted to figure out the frequency of little l, that's going to be however many L alleles we have, which is 108. I'm going to do, actually, number of L alleles divided by total number of alleles, which for us is going to be 108 of those little L's divided by 200, which is going to be, math, 0.54. So that's how you figure out the frequency, okay, of an allele if you're using a chart like this. Awesome. All right, so I do believe that that is the last thing I'm going to be doing tonight. best of luck with studying. If you have any last minute questions, I will be up for a little bit longer. So if you want to send me an email or send me a text message, feel free to do so. I would recommend doing it before about 10:30, 11, because I'll probably be unreachable after that point. And if not, then I hope you all have a wonderful night, and I will see you tomorrow for the exam.